Welcome to the SBGrid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SBGrid.
So, hello everybody, welcome to this week's SBGrid webinar series, and thanks for joining in. For today, we have Florent Delhommel. Happy to have him. He's here to talk about the research that he's been doing with the structural characterization of new co-chaperones involved in client transfer between heat shock proteins. Florent did his graduate work in Nicholas Wolf's group at Institut Pasteur, and he's currently a postdoc in Michael Sattler's group at the Institute of Structural Biology. So with that, Florent, turn it over to you.
So thank you for inviting me. And so today I will present our work on a newly identified co-chaperone that is Nazi. And this chaperone is a protein that is involved in both the Hsp90 and Hsp70 system. So just to start very generally, in the cell, one of the main functions of the cell is to maintain the cellular balance that keeps most protein into their native active state. And so there is many systems to do this. Well, one of the main one is chaperones. And what chaperones do is that they take care of newly synthesized protein or partially folded protein to bring them to their active native state. But they can also take care of misfolded or aggregated protein even and bring them back to their native state. So there is many chaperoning systems that are available in the cell, but today I will speak specifically of the Hsp70 and the Hsp90 system. So if we want to go through quickly through the system, when the protein is misfolded, it will expose hydrophobic segment that can interact more or less specifically with Hsp40. So Hsp40 will bind to the client, it will recruit it, and it will bring it to Hsp70 to form a ternary complex between Hsp40, Hsp70, and the client. And there is direct interaction between Hsp40 and Hsp70, and this direct interaction overactivates Hsp70 that triggers a cycle of folding and unfolding of the client. And so for some client, this function is enough to bring them to their folded state and to their active state. But for many stringent protein, we also need the assistance of Hsp90. So Hsp90 is a separated system, and it does not interact with either Hsp70 or Hsp40. So the first step in bringing the client to the Hsp90 system is to have a co-chaperone that interacts directly with Hsp70 and with Hsp90 simultaneously, that brings this client complex to Hsp90. And from there, the client can be loaded onto Hsp90. And from the hydrolysis of ATP, we have multiple change of conformation that eventually brings Hsp90 to its closed state, in which the client can be maturated and be folded into this active state. Now, this is a very simplified view of the system that I show you here. But in reality, these Hsp70 and Hsp90 system are very highly regulated by many co-chaperones. And especially in multicellular organism or in human, more specifically, we have over 50 co-chaperones that actually regulate either the Hsp70 system, the Hsp90 system, or connects them to each other. And this is something we're interested in the lab. We are trying to characterize this co-chaperone, but also identify new ones. And so to identify new co-chaperones, our collaborator developed a CRISPR interference screen that allows to specifically knock out individual gene linked to proteostasis. So we created around 250 cell lines that individually knock out single genes linked to proteostasis. And we then look at the effect of this knockout on the activation of GR, that is a client that has the specificity to absolutely require Hsp70 and Hsp90 for its activity. So by following the difference of activation of GR upon knockout, what we're really looking at is that the modulation of this Hsp70 and Hsp90 system induced by the knockout. So that's what you see here. In the x-axis, we have the differences in GR activation, and on the y-axis, we have the significance of these changes. And so we find some protein that we expect, like P23 or Hsp, that in blue that are non-co-chaperone of HSP90, but we also find unexpected protein or protein that we did not expect to regulate this HSP70 and HSP90 system. So we find that here, its knockout really decreased the activation of GR, but we don't know much about NOTC itself. We know its topology. It's made of a domain of a known function at its very end terminus, and then it's followed by a very long call coil. And it has been suggested that this very long call coil region induces the dimerization of NOTC in solution. So NOTC is always found as a dimer in solution, and this is suggested that it's this call coil that mediates the dimerization. We then have a CS domain that is very often found in HSP90 co-chaperones. And then we have a 60 residue region that is predicted, that is not predicted of having any secondary structure, but that is very well conserved in between organisms. So we know that the knockout of NOTC has a strong effect on G activation, but we don't know specifically how it interacts with this system. And for this, our collaborator again developed a co-IP setup coupled with mass spectrometry to identify the partner of NOTC in cell. And this is the result. What we can see is that the strongest partner of NOTC in solution is NOTC. It's NOTC itself. This is expected as the protein dimerizes. But we also see other interaction directly with the HSP40s, but also with HSP90. So we know that NOTC has an effect on the activation of client, and we see here that in cell NOTC seems to interact with HSP40 and HSP90. So just before going further on the presentation, I want to just present maybe more in detail the interaction between the protein that I'm going to talk about today. We have HSP40, that is a dimeric protein that dimerizes from its terminus. It also contains two CTD domains that are involved in client binding and in the binding to HSP70. So the very C-terminus residue of HSP70 interact with the CTD1 of HSP40. And this binding is required to free this J domain that can then interact with HSP70. So we have a 2-point interaction between 40 and 70. However, they are not independent. The J domain needs to be released by the CTD1 before being able to interact with HSP70. Now we also have HSP90. It's also a dimeric protein dimerizing by its C-terminus domain. It has 3 domains, and it has no interaction whatsoever with the HSP70 system. To connect to the HSP70 system, we need the co-chaperone Hsp that interact with the C-terminus of HSP70 and simultaneously interact with HSP90. So bringing together those two systems to transfer the client. And what we want to look at today is the position, the interaction of NOTC within this network to try to understand the function of NOTC on the folding of client. So we'll start my presentation by talking about the interaction between NOTC and the HSP70 system, so with HSP40. So we started our experiment with analytical ultracentrifugation. So we have labeled NOTC, so we are able to follow NOTC during ultracentrifugation. We can obtain the coefficient, the sedimentation coefficient of NOTC. That is directly dependent on size. And since we have NOTC label, we can then add partner in the mixture and try to see if there is increase in size that will tell us that there is formation of a complex and interaction. So for example, if we add HSP40, we see a drastic increase in size of our labeled NOTC. That tells us that NOTC in vitro also is able to interact with HSP40, as we saw in our co-IP assay. And from there, since we have a complex in which we can follow NOTC bound to HSP40, we can try to do competition with unlabeled protein and see what happens to our full-length protein. So we did this with fragments of NOTC. We just cut the protein in 2 halves and see the interaction. So when we add the C-terminus alpha of NOTC, we see that the complex between labeled NOTC and HSP40 is not disturbed at all. So the C-terminus alpha of NOTC is not able to interact with HSP40. But when we add the first alpha of NOTC, so this N-terminal region here, we do have a shift in size of our labeled NOTC. So the N-terminal part of NOTC can come in and compete with full-length labeled NOTC for the interaction with HSP40. So we know that our binding site of Nazi to HSP40 is located in this first half. So we did NMR spectroscopy on this. So this is an NMR spectrum of the first half of Nazi that you can observe here. And so for those of you that are not familiar with NMR spectroscopy, on the spectrum, each peak corresponds to a proton bound to a nitrogen. So for each peptidic bond, for each residue, we have one peak, and the position of this peak on the spectrum is directly correlated to the electronic environment of this residue. Now we can assign those peaks, we can determine which peak corresponds to which residue, and we did this for our N-terminal alpha of Nazi. And once we have this assignment, we can do a couple of things. We can determine the secondary structure of our construct. As you can see here, we can have the secondary structure propensities. So in blue, the alpha helices, and in gray, the loops. And we can see that and we can compare it to the already known topology of Nazi. So we can find that we have two alpha helices located in our domain of unknown function, but we can also see that this long region that was predicted as being folded as a coil is actually not a coil. We have actually two separated helices, a long hélice alpha 3 of 40 residue, and an hélice alpha 4 that was not predicted previously. So with NMR, we can look at secondary structure, but we can also look at dynamic. And one very easy way to look at dynamic is just comparing peak density. And if we, in our spectrum, we get much closer to the noise, we actually see that we have two regimes of intensity of peak in the spectrum. We have very high intensity peak that I showed you earlier, but we also have very low intensity peak that are located a bit earlier. And if we plot this raw peak intensity along the sequence, we see that we have two main regions. We have this region that englobes the alpha 1, alpha 2, and alpha 3 that are very low intensity. And that tells us that they behave as a very big object in the cell, in solution. So instead of having our topology with a dove and a coil, we can really see that the dimerization region of Nazi involves both this domain. It involves these three helices where the domain of unknown function works together with the alpha 3 to dimerize the protein. And we validated this with analytical centrifugation. And we can also see that our helix alpha 4 has very high intensity peak. So it tells us that this helix alpha 4 is behaving separately from the dimerization region and is more dynamic in solution. Now we can know about the topology of Nazi from NMR, but really what we want to see is the interaction with the HSP40. And for this, we did titration. So what you can see here is a spectrum where in gray we have our reference spectrum, which is just our Nazi and terminus. And in red is the same spectrum after addition of unlabeled HSP40. And what we can see is that we have many peaks that are unaffected by the addition of HSP40, but we have many peaks that have a very reduced intensity and also shift upon addition of HSP40. And that will determine our binding site for the chaperone HSP40. Now, if we plot this difference of intensity between the reference and the bound form, we can plot this intensity, and what we can see is that the dimerization domain of Nazi is not really affected by the addition of HSP40, but our independent helix alpha 4 has a drastic reduction of intensity upon addition of HSP40, which really tells us that this helix alpha 4 and this flanking region are the binding site of HSP40 on Nazi. Now we can also look the other way around. We can look at HSP40 labeled by NMR and add unlabeled Nazi on top of it. But HSP40 is a large protein. It's around 80 kilodaltons since it's a dimer. So it is too large for standard NMR. We have to use methyl labeling, specific methyl labeling, where we only look at the methyl groups of specific residues such as isoleucine, leucine, and valine. And we can still record the spectrum. So this time we're looking at the carbon in the Y dimension. We can get peaks, we can assign those peaks. So this assignment was performed by our collaborator from the Professor Rosenzweig lab. And once we have the assignment, we can look at the effect of the addition of full-length Nazi onto HSP40. So you can see on the spectrum the effect. We have peaks that are shifted like this isoleucine 203 or isoleucine 206. We can quantify those changes, quantify those shifts, and then plot them on the sequence. And what we can see is that while the J domain, the dimerization domain, and the CTD2 domain are not affected by the addition of NOTC, we have specific perturbation that happens in the linker and at the CTD1 region. And if we plot those significant perturbation on the structure, we can see that they map very well to one specific spot of the CTD1 domain. So we have determined the binding site of those two proteins from both sides of the interaction. So from there, we can produce minimal construct that contains only the rigid part that we know will interact with each other to try to do co-crystallization. So we used only this helix alpha 4 from NOTC and the CTD region of HSP40. So we co-crystallized this complex. We obtained crystal and we could solve the structure that you can observe here. We have our HSP40 in gray with the dimerization domain, the CTD2 and the CTD1. And what we can see is that our NOTC helix alpha 4 is actually bound where we expect it to bound or where we predicted the binding should occur from the NMR. But what we can also see is that in addition of the alpha 4 binding, we have this beta extension here that happens in a region that in isolated NOTC is unfolded. So we have interaction between NOTC and HSP40 that happens through this helix alpha 4 and through this beta amplification. If we look in more detail at this interaction, we can see that this helix alpha 4 interact mostly through sidechain hydrophobic interaction with the CTD1 domain. And if we look at the conservation of this residue, we can see that all the residues, so the yellow residue, pointing toward the domain are very highly conserved, while the residue that are pointing toward the solvent are not. So it really validates us that the structure that we observe in the crystal and by NMR is relevant biologically and because it's conserved between species. Now, if we look at this extra binding site that we did not expect, we can see that it goes through hydrophobic interaction, but also from hydrogen bonding. And this group of HSP40 is actually already known because this is the binding site of the C-terminus of HSP70 with HSP40. And so the sequence between NOTC and HSP70 is almost identical, at least for the residue pointing toward the domain. But while we see that the HSP70, this peptide is at the very C-terminus of the protein, so we do not have any additional binding here, but in NOTC, this is the start of our helix alpha 4 that creates our hydrophobic interaction that I just described. So we can have competition between NOTC and HSP70 for the binding of this site of HSP40. So just to summarize this part, we investigated the N-terminal alpha of NOTC. We could characterize more specifically the dimerization interface of the protein and give a function for this domain of unknown function at the very N-terminus. We could identify isolated helix alpha 4, and we could show that this helix alpha 4 is able to bind with the CTD1 of HSP40. And by solving the crystal structure, we could show that there is additional interaction that happens that comes competing directly with the binding of HSP70 for HSP40. So now that I presented you our work on the HSP40 half of the project, I want to discuss the interaction between NOTC and HSP90. And to do this, we followed a very similar pipeline to what we did for HSP40. So we started with our analytical ultrasonification assay, in which we have labeled NOTC. We can add HSP90 to it. We see the shift of size to label NOTC. And then we can do competition with our fragment. We can add the N-terminus of NOTC, and in that case, we see no shift at all in the population, which tells us that this N-terminal region of NOTC is actually not competing and is not binding to HSP90. But when we add the C-terminal alpha of NOTC to HSP90, we do see a shift of the full length label NOTC, telling us that there is competition and that the binding site of HSP90 on NOTC is in this CS C-terminus region. So we follow the same pipeline. We assign those this CS and CS C-terminus region. This is the spectrum we obtain. So we could record the NMR, we could assign them. Once we have the assignment, we can determine secondary structure. And this is what you observe here. So in orange, we have the strand propensity, and in the blue, we have the helix propensity of the protein. We can look at in the CS domain, we have mostly beta fold with some very short helixes folded. So this is expected from the general structure of CS domain and the non-structure of Nazi CS domain. But what we can see that is surprising is that this C-terminus region that was expected to be enfolded is actually folded into two large helices. And I will come back to this later on. But really, what we were interested in when we did the assignment of Nazi is to look at the interaction with Hsp90. So Hsp90 is a very large protein, is around 170 kilodaltons, so we cannot work with it by NMR directly for binding because it will just quench all our NMR signal and we will not be able to record. We will not be able to record anything. But fortunately, Hsp90 is composed of 3 separated domains that can be expressed separately. So we can express those domains individually, and we can try their interaction with the C-terminus alpha of Hsp90. And this is what you can see here. We have in gray our reference spectrum of Nazi C-terminus, and in color, overlaid on top of it, the spectrum after addition of the Hsp90 individual domain. And what you can see is that for the N-terminus domain or the C-terminus domain, once we add them, there is absolutely no change in the Nazi spectrum. However, when we add the middle domain of Hsp90, we do see a drastic reduction in intensity, and we also see some specific shift that happens upon addition of the domain. So that tells us that the CS domain of Nazi specifically interact with the middle domain of Hsp90. So we can try to characterize this interaction further now that we identify it. We can do a full titration of the middle domain of Hsp90 onto Nazi CS domain. We can follow the shifts and we can quantify these shifts and plot them on the sequence. We can identify regions that are particularly perturbated by the addition of the middle domain. And when we report them on the structure of Nazi, we can see that they form a well-defined surface that is centered around the beta 7, beta 8 strand and the beta 3, beta 4 loop. And we can see at the back of the domain, we have absolutely no perturbation. So with this NMR, with the NMR experiment, we could determine the interface of interaction between the Nazi CS domain and Hsp90. We can do the reverse titration. We also have the assignment for the middle domain of Hsp90, so to which we can add the unlabeled CS domain. And we can follow the shift, plot them, quantify them, plot them on the sequence, and map on the structure of Hsp90 middle domain the residues that are significantly perturbated. And similarly to what we saw before, we have a very well-defined interface that is centered around the alpha 2 region of Hsp90 middle domain and the beta 1, beta 2 strand. And again, no perturbation on the back of the domain. So now that we have defined those interfaces on both domains, we can try to use them to model the complex that's happening between Hsp90 middle domain and Nazi CS. So for this, we start from the crystal structure of both domains that are known, and we do rigid body docking using ADOC, in which we input our chemical shift perturbation as soft restraint. And then we can perform the docking that also follow chemical properties of proteins. And we end up with a solution that we can see here. That is our lowest energy cluster. And you can see directly that it fits very well with the interface that we determined by NMR. Now, that is expected as we introduce these chemical shift perturbation or these interfaces as an input in our docking. So we need to validate the structure. And to validate this structure, we turn to another NMR approach that is Paramagnetic Nmr, in which we chemically introduce a probe onto a cysteine of Nazi CS domain. And this paramagnetic probe has the effect of drastically reducing the intensity of the peak of the residue that are close to it in space. And the further you get to the probe, the more you recover the intensity. So we can quantify this change of intensity by comparing a reference spectrum and our paramagnetic spectrum. And this change of intensity is directly correlated to the distance between the probe and the atom observed, or the residue observed. If we look on our spectrum, we see that we have peaks in which we have low intensity, so that are close to the problem space, and peaks for which the intensity is much higher. And this can go both ways. We can use the intensity change to directly determine the distance between the probe and the residue, but we can also, starting from the distance in our model, predict the paramagnetic effect that we expect for our complex. And this is what we did. So in purple dot is our measured PRE, so paramagnetic changes of intensity from the NMR experiment. And in green is our calculated PRE from the model. And as you can see, we have very good match, we have very good correlation between the intensity reduction that we observe in our experiments and the intensity reduction that is expected from the structure. So by using Paramagnetic Nmr, we can validate the model that we generated using chemical perturbation. Now, we have created this model on the isolated domain, but we really want to validate it and validate that this interaction is actually happening on the full-length protein. So for this, we want to look at Hsp90, but Hsp90 is very large. We need to use, again, this specific labeling. So we specifically label the isoleucine or the methyl of the isoleucine on Hsp90. That gives us around 50 probe well spread all over the structure. We can record a spectrum of this, and we can assign it. We can determine which peak corresponds to which isoleucine. And once we've done this, we can do interaction. So we can add Nazi to this, and what we can see is that we have a specific binding. We have specific perturbation on our spectrum, while other peaks are not affected by the addition of Nazi. Now, if we quantify this and plot this on the sequence, the first thing that we can see is that our N-terminus domain is absolutely not perturbated by the addition of Nazi. This validates what we observe with single domain. Nazi does not interact with the N-terminus domain of Hsp90. But what we observe is that most of our perturbations are located around the middle domain of Hsp90. And if we report this on the structure, we see that most of the perturbation happens around this alpha-2 helix and beta-1, beta-2 that we identify as the binding site of Nazi onto Hsp90. So that really validates the model that we made on the isolated domain. But as you can see, we also have extra perturbation that are located here, that we can see on the plot here, that are located in between the middle domain and C-terminus interface that I will call MC interface, starting from now. So we have these extra perturbation that are visible that we did not expect at the MC interface and that we did not observe in the single domain experiments. So to characterize further this binding, we did some additional experiments in which we used fragment of Nazi and repeated this binding to full-length Hsp90. This is what you can see here. We have our full-length binding as a reference on the top, and then we perform the same binding with the C-terminal region of Nazi. And what you can see is that we have identical perturbation. We have an identical pattern of perturbation using only our CS C-terminus of Nazi compared to our full-length binding. So that tells us that the CS terminus is alone responsible for the binding to Hsp90 and is able to recreate the pattern of the full-length binding. We also did an extra experiment in which we used a fragment of Hsp90 that just misses those two C-terminal alpha helices. And what we observe in that case is that we still have perturbation, we still have binding between Nazi and Hsp90. However, we do not have the same exact pattern. We still have a very strong effect on those residues that are located around the alpha-2. So that really confirms us that this binding that we observe in the full-length binding is the binding of the CS domain to Hsp90. And that shows us that this CS domain is enough to create a strong binding to Hsp90. But we observe that in this truncated form of Nazi, we misses those extra effects that we observe at the MC interface. So with this, we can conclude that this terminal, even though the CS domain creates the main binding to Hsp90, these terminal helices can come and create additional binding at the MC interface. So since we validated that the main interaction between Hsp90 and Nazi happens between the CS domain and the middle domain of Hsp90, we just align this model onto the full structure of HSP90. So it's just an alignment, it's not a model or not a structure, but we can still conclude a couple of things, or at least compare this model from what we know experimentally. The first thing that we can see is that our CS domain in the full length alignment does not contact the NTD of any of the two protomers. And it's something we know, the CS domain does not interact with the NTD. But we also see that the CS domain does not contact with the other protomer of HSP90. And this is something that we know from biophysics. The Nazi binding does not interfere with HSP90 closing or ATPase activity. So it tells us that Nazi does not contact, and this is what we see with our model. Now, the other thing that we can conclude from this alignment is that if we look at the C-terminus of our CS structure, it localizes exactly at the bottom of the middle domain. And that will put our C-terminal helices exactly in the right spot to come and create these extra MC interaction that we see from the NMR. So with this model, with this alignment of our model, we really can, or at least it fits very well with our experiment. So now to summarize what we observed on HSP90, we could again refine the topology of Nazi by defining these C-terminal alpha helices. We could show that the main interaction between HSP90 and the CS domain and Nazi happens from the CS domain and the middle domain of HSP90, and we could model this complex. And we could also show that there is some extra interaction that are not essential or for the binding between the C-terminal helices and the MC interface of HSP90. Now, I've shown you the interaction of Nazi between HSP40 and HSP90 individually, but we also want to validate that Nazi is able to bind to these two proteins simultaneously. And for this, we methyl label our Nazi protein. So it gives us just a few probes on the sequence. We label the isoleucine. That gives us 2 probes on the dimerization region of Nazi, 2 probes in the HSP40 binding site, and a few more in the binding site to HSP90. So, and then we can create interaction with only HSP90, but also with a mixture containing HSP40 and HSP90. And now we can quantify the changes that happens upon this interaction. So if we compare our reference spectrum before and after addition of HSP90, we see that we have intensity reduction mostly localized in the CS domain. This is what we know. This is the binding site of HSP90 on Nazi. But if we add HSP40 to this already preformed complex, we see a drastic reduction of intensity on this helix alpha 4 without affecting the intensity in the CS domain. So that really proves us that Nazi is able to interact simultaneously to the 2 regions that we define as interacting with HSP40 and with HSP90. Now, I would like to use the last part of this talk to try to describe the interaction between Nazi, the effect of Nazi on client refolding. And so in that case, we went back to analytical intercentrifugation, but this time, instead of labeling Nazi, we labeled the client GR. And from there, we can try to investigate the fate of the client upon addition of various mix of chaperone and co-chaperone. So for example, as you can see here, when we have our client, we can add HSP40 to it. We create a complex that we can observe at this sedimentation coefficient size. If we add HSP70 on top of this, we create an even bigger complex that involves HSP40, HSP70, and the client. So this is all known and expected. But if we add Nazi on this mixture, what we observe in that case is a decrease in size. So it tells us that the client GR is bound to a complex that is smaller than the one observed with HSP70. And if we try to investigate what this complex is, it fits perfectly with a complex that only includes HSP40, Nazi, and the client. So what we think happened in that case is that we have our ternary complex, client complex that involves client, HSP70, and HSP40. And when Nazi comes in, it excludes HSP70 from this complex. And this is something we know from the crystal structure. There is competition between Nazi and HSP70 for the binding to 40. And once HSP70 is excluded, we create a complex that includes HSP40, Nazi, and the client. Now we can add more chaperone to the system. We can look at what happens with Hsp90. So this green curve here is the complex that is the different complexes that are formed when GR is labeled and Hsp40, Hsp70, and Hsp90 are added. What we can see is that we obtain two different complexes, two different sizes that correspond to our ternary complex between the client, Hsp40, and Hsp70. And this earlier peak that we observe here is the complex that is formed directly between the client and Hsp90. But as I presented you earlier, there is no direct contact between those two systems. So the client cannot form a large complex with them too. It's either bound to Hsp90 or bound to the Hsp70 complex. And when we add Nazi to this mixture, what we observe is a single peak, a single species that is smaller than the Hsp70 complex. And if we try to characterize this complex, it fits exactly to what we expect for a complex that involves Hsp90, Nazi, Hsp40, and the client. So if we want to summarize this, when we start with our mixture of clients bound to Hsp70 or to Hsp90, when Nazi comes in, we see the similar effect as I presented earlier. Hsp70 gets excluded. Nazi is able to bind to 40 and the client. And since Nazi is able to interact independently to Hsp90, it brings it to the final Hsp90 complex and creates a full complex in which Nazi is able to bridge Hsp40 to Hsp90 while still holding the client. So just to summarize our findings, we started from a well-known and well-identified pathway to load the client from the 70-40 complex that involves this co-chaperone hop that can interact simultaneously to Hsp70 and Hsp90. But today what I showed you is that by investigating Nazi, we could show that this protein can bind directly to Hsp40 and the client and bring this client complex to Hsp90. And what we could further show is that this Nazi protein is capable of displacing Hsp70 from the client complex and bring this 40 client complex to Hsp90. And once this is loaded, when this is loaded onto Hsp90, the client can be loaded on Hsp90 and brought back through the change of conformation of Hsp90 to its mature conformation. And so what we really showed with our analysis of Nazi is we propose, or we have shown an alternative pathway of loading the client onto Hsp90 that does not involve the co-chaperone hop. And with this, I would like to thank all the people that were involved in this project. So of course, Sattler that supervised me for my postdoc studies, and all the people that have been involved in the NMR study and the crystallography study that I presented. Now, this project is always developing close collaboration from the Professor Buchner lab that are specialists of Hsp90 and that has performed all the experiments that involve biophysics of these complexes. We also collaborate with the Professor Rosenzweig and her lab has performed all the experiments that involves methyl labeling of Hsp40. And we also have some additional collaboration for the CRISPR screening with the Professor Kamban lab and Professor Lam lab for threat experiments for the folding of client that I did not present today. And I would like to thank you for your attention, and I will be happy to answer your questions.
Thanks, Florent. That was a very good talk, and I have questions, and for folks in the audience, if you have questions, please feel free, send them in chat, or if you'd like to raise your hand and unmute yourself, you can do that as well. So I guess just to get started with the Nazi dimer and the Hsp40 interactions.
Yes.
So there was one of your earlier slides where you were showing the dimer interface mapped on the Hsp40 with the Nazi interaction domain. Is that something that's spatially consistent with Nazi forming a dimer?
Yeah.
And I apologize that if the question is something that was not, if I did a bad job asking the question, let me know.
No, that's fine. So we have the alpha-3 helix that starts fairly close to this region. So we could have the alpha-3 bound here. So this is consistent. Now it is difficult to. I mean, we could not. It's very difficult to determine if we have a 2-to-2 binding. When we do, what I can tell you is that when we do this experiment, we do not see splitting. So we have a single population that is observed shifted. So it would tell us that both sites of HSP40 are occupied. So it seems from the NMR that both sites are occupied at the same time by the addition of Nazi. Now, I didn't look in detail if we have space. We have a few residue of linker before the start of our alpha-3 helix that is very long and that also has some flexibility. But we have the NMR data. There is some spacing, but I don't have a definite answer for you.
Yeah, case. Well, you can model, and if it's consistent, but it may not be worth doing the experiments to get it nailed down for that. And also, if there was something odd with you'd probably see that on the columns or the centrifugation plots. So.
Yes, so then that will start to create polymers, I guess, with the image one, which we didn't see. We have seen population.
So another question, you followed for Nazi and Hsp40, you had the very good sequence with NMR to isolating the domains to crystallizing to confirm the domains. Mm-hmm. And then for Hsp90, you took a different path with swapping out the crystallization for the haddock docking. Was that was that just for practical reasons because you tried crystals and they didn't work, or was there a different reason you chose to go that route?
Yeah, I mean, it just didn't crystallize. The affinity between the isolated domain is fairly low. So since those two proteins are dimeric, once we have the full-length protein, the KD is actually around 600 nanomolar. So the 2-to-2 binding between the two proteins is very high, but the isolated domain without the avidity that brings the dimerization is fairly low. So we could not crystallize the complex. So that's why we did the docking plus validation. Of course, if we could have had the crystal structure, that would have been better.
Yeah, well, I'm always curious about things like that. For where would somebody have tried one technique versus another technique, and yeah, but sometimes because the one you wanted to try didn't work, and but sometimes the reason for asking is because sometimes there's a reason other than that. So
no, we always try to go with any technique that is the best to answer our solution, but in that case, the crystallography did not work.
And this talk was a great example of combining techniques from multiple different areas to get answers to the biological questions you're trying to go to.
Thank you.
And we have a question from Maru. And Maru, if you'd like to unmute yourself, please feel free. Otherwise, I'll read that off.
Yeah, wonderful talk, Florent. So Nazi appears to compete off Hsp70.
Yes.
Like you said, 600 nanomolar is a really tight interaction. What do you hypothesize Nazi's function to be? Like, have you had any work showing like GR's function or like the actual chaperoning ability?
So, I mean, we think that the real function is really this transfer from 40 or at least 40-70 complex to Hsp90. We do have some functional analysis on the client I did not present. So we can look at the activation of the client basically by following the binding to hormone. So we can start from the unfolded system with 70 and basically what we very quickly, but if we only add Hsp90, we don't have reactivation of the client. But if we add Nazi by itself, it reactivates the client. And we think that's because it kicks out the 70, which then cannot keep the client unfolded, and that will refold the client. And then to get the extra bit of reactivation, we need the transfer to Hsp90. We need the presence of Hsp90. So we really think that the function of Nazi is to transfer the client to Hsp90 to finish the folding.
Thank you.
And we've got another question from Genevieve Seabrook. So if you've got your hand up, so if you'd like to unmute and ask, please do.
Hi, very beautiful presentation and beautiful work from very interesting stuff. I was just curious to know whether you were able to measure the binding affinity of Nazi and Hsp90.
Yes, so this has been done by ultracentrifugation. I do have it somewhere. But yes, so this is around 600 nanomolar. But again, this is for the 2-to-2 binding. So there is a lot of avidity going on. When we look at the single domain binding, we are more in the 50 micromolar also. But the full length protein, this is around 650 nanomolar.
Okay, thank you.
So I guess one other question is for the CTD, the Nazi CTD Hsp90 interaction. Is that something where it's robust enough that you're distinguishing the weak interaction with the middle domain in CTD versus conformational changes?
Yes, so that's always very difficult with Hsp90 because we have a lot of conformational changes that can be induced by the client. But in that case, I think we are very lucky that we have those fairly specific effects on the methylable spectrum. And we know also from the biophysics that, so we have pretty nice effect on the middle domain that we can characterize. And we know from the biophysics that the presence of Nazi, unlike many co-chaperone, does not really affect, is it this one? No. Anyway, basically Hsp90 doesn't, no, sorry, Nazi does not really affect the conformational cycle of Hsp90. So the presence of Nazi does not change the ATPase hydrolysis, it does not lock the Hsp90 in a closed conformation and so on. So we don't really have, and this is what will trigger the allosteric effect. So Nazi seems to not really influence Hsp90, which alleviate all these allosteric effects that we could see and that would really pollute our NMR spectrum.
If we don't have any more questions, then I guess thank you again for joining us and for presenting your research.
Yep, thank you very much.
And for doing it, because it looks like it's very cool stuff.
Thank you for inviting me.